Good morning, grade 9 students. Our lesson today about from food into nutrients digestion. The objectives of this lesson: define digestion, differentiate between organic and inorganic food, define each group in organic food and give example for each group, identify experimentally the presence of each group in any food. This figure shows the digestive system of a human. This digestive system composed of digestive tract and the digestive glands. The digestive tract or the digestive tube includes all the digestive organs where the food passes, starting from the mouth, esophagus, stomach, small intestine, large intestine, and the digestive glands, salivary glands in the mouth, liver, and pancreas. Then, as a definition, the digestion is the process during which large food molecules are broken down into smaller ones and finally into nutrients. Digestion is the set of mechanical action and chemical action. Mechanical action includes all the movements like chewing the food in the teeth, mastication, churning, and peristaltic movements in the esophagus. Chemical action is the secretion of digestive juices by the digestive glands. Example, saliva secreted by the salivary glands in the mouth. Also, the gastric juice is the juice secreted by the glands in the stomach. Intestinal juice is secreted in the intestine. Pancreatic juice is secreted by the pancreas. Now, in activity 1, which is our food. Food can either be organic food or inorganic food. Organic food such as carbohydrates, proteins, lipids, vitamins, whereas inorganic food such as water and mineral salts. The difference between organic food and inorganic food is that organic food contains carbon, whereas inorganic food doesn't contain carbon. Starting from the first group of organic food, which is the carbohydrates. Carbohydrates are energetic food. It gives energy to our body. It includes 3 classes: monosaccharide, disaccharide, polysaccharide. Monosaccharide, it is simple sugar molecule. Example, glucose. Whereas disaccharide, it is made up of 2 sugar molecules linked together. Example, maltose. Maltose, it is made up of 2 glucose molecules. Whereas polysaccharide, example, starch and cellulose, it is many sugar molecules linked together forming polysaccharide. Here are some examples about carbohydrates, like in the adjacent figure. Bread, pasta, beans, rice are energetic food. They give energy to our body. The second group in organic food is the protein. Proteins are the building blocks of the body. They are builders' food. Example of a proteins: meat, fish. Proteins is like a chain. Like in this adjacent figure, it is made up of simplest unit, which is called amino acid. Protein is this chain, and each one in this chain, it is called amino acid. So proteins is made up of amino acid linked to each other, forming this chain. The third group of organic food is lipid. Lipids are also energetic food. It's made up of 1 glycerol molecule and 3 fatty acid molecules. This football is the lipid molecule. This lipid molecule is made up of black one, which is the glycerol, and this black one, it is surrounded by 3 white ones, which they are the fatty acids. So lipids is made up of 1 glycerol molecule and 3 fatty acid molecules. When the lipids are digested, it becomes 3 fatty acid molecules and 1 glycerol molecule. Example of lipid: oil, butter. These groups of organic food, they can be identified using some tests. For example, iodine test is used to identify the presence of starch, which is the carbohydrate. It gives dark blue color after dropping few drops of iodine solution on a food, for example, potato, like in the adjacent figure. It gives dark blue color in the presence of starch. If there is no dark blue color, so there is no starch. The second test is Fehling Test, which is used to identify the presence of reducing sugar, which is also carbohydrate but is sugar, not the starch. The Fehling test should be done in the presence of heat after dropping a few drops of Fehling solution. In the presence of heat, it gives a precipitate. The third test is a Biuret test. It is used to identify the presence of a protein. It gives positive result, or it gives a violet color in the presence of a protein. If there is no protein, so there is no violet color. After putting few drops of copper sulfate solution and few drops of sodium hydroxide solution, which both make Biuret reagent, it should give violet color, and this violet color means that there is a protein in this food. The last test is a paper test, which is used to identify the presence of lipid. After putting some drops of oil, for example, on this paper, translucent spot appears, and this means that there is lipid.